All right, so this is a brain dump. We're proving this can be done in 20 minutes. Get ready, get set, go. There are 4 types of biomolecules. Lipids, carbs, proteins, nucleic acids. Lipids come from fats. Carbs are basically sugars, other things too. Proteins come from meats, also beans, and nucleic acids come from the nuclei of cells. Their shapes help them do things. The cell membrane, the glucose hexagon, the beads on a string that fold into shapes for proteins, DNA that we've been learning about all year. I made a really bad DNA right there. And then what do? Lipids form the cell membrane. They also do energy storage. Carbohydrates give us quick energy. Proteins are most important for enzymes, but also fingernails, hair, enzymes, and nucleic acids give genetic instructions. Protein synthesis happens in 2 stages. Transcription happens in the nucleus. Translation happens in ribosomes. Ribosomes are important. So we've got our DNA and it's got to open up. And as it opens up, we have these nitrogenous bases, A, T, C, and G. The messenger RNA is going to carry that out of a nuclear pore, messenger RNA. And let's remind ourselves what connects up. A would normally bond with T. It can't bond with T. It has to bond with U because messenger RNA doesn't use T. But T still goes with A, C goes with G, G goes with C. This all happens in the nucleus, comes out through a nuclear pore. And then it's fed into the ribosome. And the ribosome reads these 3 at a time to make this chain of amino acids we call a protein. It's nice to identify that this is a protein, to remember what it is that's being built. The ribosome looks like a hamburger. And then, how does this go out from the cell? It starts in the nucleus. It comes out. Here's our messenger RNA. Thank you. Messenger RNA. It gets read by a ribosome that's on the endoplasmic reticulum. Endoplasmic reticulum. The protein goes through here. It can come out through a vesicle. This was the nucleus. It can go into the Golgi apparatus, which looks similar to the endoplasmic reticulum, except it's near the cell membrane. And then this here is supposed to represent exocytosis. Right, and so these 2 processes here, that's what's happening in here. 1/3 of the way done. There's passive and active transport. And then leave yourself some room over here to talk about mutations, separate thing. Most basic form of passive transport is diffusion. It's when you have a high concentration outside the cell, a low concentration inside, and things naturally go in. Facilitated diffusion, uses proteins in the cell membrane to get things in. So this is facilitated diffusion. I'm just going to be quick and just say facilitated. For active transport, you have to use ATP to move things from a low concentration to a high concentration. These are obviously cell membranes. I'm not putting the double layer of the phospholipid bilayer. For mutations, the best way to remember how mutations work is pretend we had a section of DNA that had the code cat. Since DNA is read 3 letters at a time, this is called a codon. This codes for histidine. Same with this one, histidine. But if we change it up and add an extra letter, now it says cat, a cat. Since these are read 3 at a time, now instead of CAT we have ACA. This codes for threonine. And that's going to be very different. So what I want you to remember is that insertions and deletions cause frame shift. All right, I'm writing with bad handwriting because I'm trying to go quick. How does DNA replicate itself? First, we have DNA helicase, which unzips the DNA. And then we have 2 molecules of DNA polymerase that builds the new strands of DNA. These actually work in opposite directions, but that's here and here. 4 phases of mitosis. Not 4 phases, 4 places plus cytokinesis. So pass me taco. Chef stands for prophase, metaphase, anaphase, telophase, and cytokinesis. Prophase, the nucleus disappears and dissolves. The DNA condenses into chromosomes. In metaphase, the chromosomes line up at the midline, and spindle fibers form from centrioles that are going to reel those in. They sort of have a right angle shape to them, like 2 cans of soup at right angles. Anaphase, they start to pull apart, flapping in the breeze as they do. So those get reeled in like a fishing line and fishing reel to those centrioles to the 2 sides of the cell. In telophase, we have what's called cleavage. We have 2 nuclei reappearing, and the DNA unwinds from chromosomes to make that DNA shape again, so it can start being used. And in cytokinesis, the 2 cells are separate. If you want to, you can just leave them both like that. Just to be quick, we'll put in the DNA inside the nucleus. In plant cells and skin cells, these are still touching. In amoeba or paramecium, they swim off in different directions. For meiosis, this is not exactly right, but it's a good way to help think about it. Number of chromosomes is what we're dealing with here. This is for a human. 46 is what we start with in a normal cell. Sorry, right here, number of chromosomes, 46 in a normal cell. We duplicate the DNA. That's just what we saw up above. Up here, we duplicate the DNA. And then the cell divides, just like it would in mitosis, giving 2 cells with 46 chromosomes. But then in meiosis, it divides again. So this is 23, 23, 23, 23. These would be sperm cells or egg cells, pollen or caviar if this is fish. Again, this is not actually exactly what's happening, but it's pretty close. It's worth for us to remind that we're making haploid cells right here, and these are designated 1N, and that up in mitosis we're making diploid cells, which are designated 2N. 2 copies of each chromosome, 1 inherited from mom, 1 inherited from dad. All right, 2/3 of the way done. Prokaryotes versus eukaryotes. Prokaryotes have no nucleus. An example of them would be bacteria. Eukaryotes have a nucleus. They also have membrane-bound organelles. They both have DNA, they both have ribosomes, and they both have cytoplasm. So we can make a Venn diagram of these two. Let's remind ourselves about viruses, which are not alive. And they need to infect a host cell to replicate. It's worth noting viruses do not have cytoplasm or ribosomes. All right, photosynthesis. This happens in chloroplasts. That's fine. We can abbreviate. Cellular respiration. Respiration happens in mitochondria. Plants need sunlight. Plants need water. That's H2O. And then let's say you can't remember what that is. You know that plants make oxygen, and they can give us fruits that have sugar. Technically, this is C6H12O6, glucose, blood sugar. If plants make oxygen, that means that they needed to breathe in carbon dioxide for their chloroplasts. And then cell respiration is most of this just reversed. Or just our mitochondria, they use sugar. We already said what that was. And we breathe in oxygen, and we breathe out carbon dioxide, and we have to urinate. We make water, which is H2O, and this process also produces ATP, cellular energy. Photosynthesis, cellular respiration. Genetics. Remember the difference between phenotype and genotype. Phenotype is what it looks like. It's the phenomenon. And we use the example of having brown eyes or blue eyes. Thank you. Lowercase b. And then the genotype is the alleles. And if we put it in this form, 2 lowercase, 2 uppercase, and then 1 of each, we can remind ourselves that those are homozygous. This is heterozygous. Then we have dominant and recessive. Heterozygous is 1 of each. And then this is the big important idea. Just one dominant allele gives dominant trait. If you know that, everything else you can figure out. If you don't know that, it's hopeless. All right, and then let's remind ourselves how to do a dihybrid cross. I suggest we continue to use the same example I've been using, but it doesn't matter. All you need to do is remember to use the FOIL method. Again, we're not going to solve this. We're just going to set it up. If you can set it up, solving it's easy. So let's zoom in. So the FOIL method. First, outer, inner, last. Same thing over here. First, outer, little a, little g, inner, little a, big G, last. You can do the rest from here. Remember to analyze it. Everything in here will have 4 letters because we're analyzing 2 traits at the same time. Okay, nearly done. Let's talk about ecology.
Cut off.
Thank you. Parasites. Like mosquitoes, suck your blood. Great for them, not so great for you. Commensalism is when somebody gets something good, somebody else gets nothing. A great example of this would be the remora fish, which has that suction cup head, gets a free ride from sharks, doesn't hurt the shark. Mutualism is when both get something good. This would be like bees and flowers. Bees get a free meal, they get food, nutrients. sugars from the plant, the flowers get to do sexual reproduction. They get pollination. Okay, and then vocabulary you're likely to forget. Autotrophs are plants. Xylem carries stuff up from the roots. Phloem carries stuff both directions. Heterotrophs. Oh, let's not forget thigmotropism. Thigmotropism, growth in response to touch. Hard one to remember. T and T can help. Our example would be ivy. I-V-Y, ivy. And then our heterotrophs. This would be animals. They eat others. That's what the name means. And let's not forget that the endocrine system uses hormones. That the integumentary system is the skin. And that the excretory system is things like the kidneys. It produces urine. Okay, 16 and a half minutes. And we just made ourselves a cheat sheet for 80% of the Texas Biology STAAR test. Just for the purposes of this video, I'm going to go through. We're going to look at these.
Is that all?
So this is our third page. That's it. This was our second page. Hold on. And then this was our first page. Next time you do this, you'll do it from memory.